Hello, dear students. I welcome you all on behalf of Sri Jayendra Rank Zone classes. Dear student, we are in a continuation of the chapter that is respiration in plants. In few previous lecture, we have already talked about the introductory part, the glycolysis, the difference between aerobic and anaerobic respiration. And in aerobic respiration, we have also discussed the fate of pyruvic acid, where we have discussed the link reaction and the Krebs cycle. Now, till Krebs cycle, in glycolysis, link reaction, and Krebs cycle, we found a synthesis of NADH2 and FADH2. And I told you to get a 3 ATP from NADH2 and 2 ATP from FADH2. There is a need to, there is a need of a ETS happen. So we are going to discuss here electron transport system or electron transport chain and oxidative phosphorylation. In this, the electron transport system and oxidative phosphorylation, several electron carriers are located in the inner mitochondrial membrane. And these several electron carrier which are located in the inner mitochondrial membrane play a role in a transport of H plus from the mitochondrial matrix to the perimitochondrial space. See, this is the inner mitochondrial membrane. On outer side of this, you will see the outer mitochondrial membrane. This the between the outer and inner mitochondrial membrane, you will see the perimitochondrial space. And these are the complex 1, this is complex 2, this is complex 3, this is complex 4, and this is complex 5. Okay, like these complexes are involved in the inner mitochondrial membrane, they help in carrying out a electron transport system, and these complexes play a role in transfer of H plus from the mitochondrial matrix to the perimitochondrial space, and this H plus help in a synthesis of ATP. We will discuss how. Okay, so this is the thing. Now here the proteins are involved. So these all the 5 complexes are the nothing but a protein complexes, and they play a role in electron transport chain. So series of proteins in the mitochondria help in transfer of electrons from NADH to oxygen or FADH to oxygen and gaining of ATP. So most energy from redox reactions, if we get the electrons during the metabolic reaction sent to NAD and FAD, and due to this, most of the redox reaction, the electrons from the metabolic reactions are sent to NAD and FAD, and the NAD will form NADH2 and FAD will form FADH2. And number of NADH and FADH2 produced at various levels of aerobic respiration. This number of NADH2 and FADH2, which are produced at various levels of aerobic respiration, they help in gaining of ATP or energy. So electron transport system involves 5 multi-protein complexes. We will discuss that which 5 are there, 1 to 5, and we have just seen. And in that we will discuss that 2 mobile electron carriers are there, ubiquinone and cytochrome C. So first we will discuss how NADH2 is equal to 3 ATP. Okay, that we need to discuss. If we discuss that in mitochondria, as we know, there is a outer mitochondrial membrane. This is outer mitochondrial membrane. And here we see the inner mitochondrial membrane. Now inner mitochondrial membrane fold and form a crystal like this. So this is outer mitochondrial membrane. This is inner mitochondrial membrane. And this is the space between outer mitochondrial membrane and inner mitochondrial membrane. That is a perimitochondrial space. Okay. Now we know that this is mitochondrial matrix, and in mitochondrial matrix, TCA cycle or Krebs cycle take place. And during TCA, we know that NADH2 and FADH2 are produced. Now NADH2 and FADH2 which are produced during the TCA cycle, in this if you talk about the first NADH2, then NADH2 break into the NAD plus 2H plus and 2 electron. Now this 2H plus and 2 electron which is coming from the NADH2 breakdown, that is accepted by FMN. It is also one electron hydrogen carrier. When FMN receives the 2 electron and 2H plus from the NADH2, it forms a reduced FMN, that is FMNH2. Okay. Again, when FMNH2 break or oxidize, is the FMN is received back. The 2H plus are passed to the perimitochondrial space and 2 electron are further transported to the iron sulfur protein, which is the electron carrier. From iron sulfur protein, 2 electron passes to ubiquinone. It ultimately passes to the ubiquinone. Then, when ubiquinone receives the 2 electron, this accepts 2H plus from the mitochondrial matrix and converted into UKH2. When UKH2 break, it forms ubiquinone again. 2H plus are passed to the perimitochondrial space and 2 electrons are given to cytochrome B. Okay, cytochrome B. Now cytochrome B gives its 2 electron to iron sulfur protein. Iron sulfur protein gives its 2 electron again to ubiquinone. This ubiquinone again receives 2H plus from the, we can say, the perimitochondrial matrix and converted into UQH2. When UQH2 again get oxidized, ubiquinone will be obtained. 2H plus are moving to the perimitochondrial space and it passes its 2 electron to cytochrome C1. From cytochrome C1, the electron passes to the cytochrome C, which is located on the outer side of an inner mitochondrial membrane. Then its cytochrome C passes its 2 electron to cytochrome A. Cytochrome passes its 2 electron to cytochrome A3, and this cytochrome A3, when receives 2 electron, it take half O2 from the matrix, 2H plus from the matrix, and 2H plus and half O2 react and form H2O. Okay, so H2O ultimately formed by the reaction of the half O2 2H plus from the mitochondrial matrix and the electron which is received from the cytochrome A3, which was ultimately transferred from the NADH2, and ultimately it is received by the O2, and then water formation occurs. So when NADH2 breakdown occur and the electron passes through this many electron carrier, ultimately it reaches to the half O2 and water synthesis occur. And during this electron transport, the 3 pairs of H plus are pushed to the perimitochondrial space. Now it is known that here to push this, here this is the complex 1 which is involved in particular, this is known as complex 1. After this, in NADH2 background, cytochrome BC1 is known as complex 3. This cytochrome A3, it is known as complex 4. Okay, so complex 1 and complex 3 and complex 4 are involved in the electron transport from NADH2 to the oxygen and during the NADH2 breakdown. Now what happens in the cristae, there is a F0 and F1 complex. Now this F0 complex and F1, if we talk, F1 is present in the inner mitochondrial membrane and F1 is protruded towards the mitochondrial matrix. Now when one pair of 2H plus passes from F0 to F1 or through the F0. F0 is the integral protein through which when the one pair passes, it passes its electron to F1, and that energy to F1, that energy used by F1, and F1 cause a conformational changes in it, and it carry out a condensation of ADP and inorganic phosphate. So due to the condensation of ADP and inorganic phosphate, the ATP ultimately going to synthesize. So when one pair passes through the F0 and releases the energy that used in the condensation of ADP and inorganic phosphate, and one ATP is synthesized, when the second will move and then third pair will move through this F1, ultimately three pairs of ATP is going to be synthesized. Okay, when three times three pairs of proton are pushed moved from F0 to the mitochondrial matrix, the energy released three times, and three times this energy is used in the condensation of ADP into inorganic phosphate. So when three ADP will condense with the three inorganic phosphate, and three ATP are going ultimately synthesized due to this passing of proton back to the mitochondrial matrix. Okay, so this is the thing. Now, now how NADH2 gives 3 ATP that we understood. Now, what are the electron carrier in this? We remember one FMN is there, okay, one ubiquinone is there, okay. What are the full form of them? I will enlist them, and what are the complex 1, complex 3, and complex 4? What names are given? That all we will discuss in the later part. So this is how NADH2 give rise to the 3 ATP by its breakdown. But when FADH2 breakdown occur, ultimately 2 ATP is going to be synthesized. Before that, we should note down some points. FMN is flavin mononucleotide, and when we write FMNH2, we call it as reduced FMN, that is flavin mononucleotide. Apart from this, UQ, that is ubiquinone, is also known as coenzyme Q. So we can write it as a ubiquinone, or we can write call it a coenzyme Q. Okay. Then after this ubiquinone coenzyme Q, next if we write, then we write a reduced ubiquinone or reduced coenzyme Q. So we can write it as a reduced ubiquinone, and this reduced ubiquinone is also called as ubiquinol, and it can also be called as reduced coenzyme Q. Reduced coenzyme Q. Such a way this particular thing happens. Now, after this, ubiquinone and coenzyme Q equals to, that is reduced ubiquinone. Apart from this, one more thing is that cytochrome C. CYT stands for cytochrome. In this B, remember, we have seen one cytochrome C. This cytochrome C, if we talk, it is the one mobile electron carrier, and it is located on the outer face of inner mitochondrial membrane. So we can write that this cytochrome C, it is a small protein attached to outer surface of inner mitochondrial membrane. Attached to outer surface of inner mitochondrial membrane, and it act as mobile electron carrier. Okay, so this is the thing which happens during this, that is the cytochrome C. Now, after this, we have to discuss how FADH2 breakdown occur. And to understand FADH2 breakdown, we have to draw here again outer mitochondrial membrane, and we know how this FADH2 give rise to 2 ATP that we have to understand. So outer mitochondrial membrane is there, perimitochondrial space is there, and inner mitochondrial membrane is there. Now here this is mitochondrial matrix, and we know that Krebs cycle take place here, and FADH2 and NADH2 are synthesized here through Krebs cycle. Now when FADH2 undergo breakdown, FADH2 break into FAD plus, and along with FAD plus, it is known that this gives its electron to iron sulfur protein. 2H plus and 2 electron are given to iron sulfur protein. After iron sulfur, it gives its 2 electron and 2H plus to ubiquinone. So this ubiquinone get converted into UQH2. UQH2 break into ubiquinone, 2H plus, then 2H plus passes to the perimitochondrial space, and the 2 electron are given to cytochrome B. Then cytochrome B gives 2 electron to iron sulfur protein, then 2 electron to ubiquinone. Ubiquinone accepts 2H plus and converted into UQH2 again. UQH2 when again break give rise to the ubiquinone and 2H plus, which goes to the perimitochondrial space. The 2 electron are passed to cytochrome C1. From cytochrome C1 it reaches to the cytochrome C. Okay, so here ultimately the electron are passes from cytochrome C1 to the cytochrome C, and as we have discussed, cytochrome C is present on the outer face of inner mitochondrial membrane. Now it passes its two electron to cytochrome A, then again it passes 2 electron to cytochrome A3, and when it gives 2 electron to the cytochrome A3, it accepts half O2 from the mitochondrial matrix, then 2H plus from the mitochondrial matrix, and ultimately the water is going to be synthesized. And like this, the due to the breakdown of FADH2, the electron ultimately transferred to the terminal electron acceptor, that is half O2, and water is getting synthesized. And during it, in the transport of electron, here this complex 2 is involved. Then cytochrome B C1, that is complex 3 is involved, and cytochrome A3, that is complex 4 is involved. Okay, and like this ultimately the during the transfer of electron from FADH2 to the half O2, the two pairs of proton are passes to the perimitochondrial space. So as we know here, there is a F0 F1 complex. As the one pair comes back, this one, then one ATP getting synthesized. But if two pairs are there, these two pairs come back to the mitochondrial matrix to the F0, then F1 carry out a synthesis of 2 ATP. And because of this, we call FADH2 is equal to 2 ATP. Okay. The difference is complex 1 is involved in NADH2 breakdown, but in FADH2 breakdown, complex 2 is involved. Complex 3 and complex 4 are common in both them. Okay. So now we are getting it. Why FADH2 and NADH2 is calling, particularly we can say the two FADH2 is equal to 2 ATP and NADH2 is equal to NADH2 is equal to 3 ATP and FADH2 is equal to 2 ATP. Why it is calling, that we have now understood. So now we are clear that in NADH2 breakdown, or oxidation, complex 1, 3 and 4 are inward. Whereas during FADH2 breakdown, complex second, third and fourth are inward. Means the complex 3 and 4 are common for both breakdown getting ATP, but complex 1 is exclusive for the NADH2 breakdown and complex 2 is exclusive for the FADH2 breakdown. Now, as we have seen here, there are 5 complexes in this. As we have seen, ubiquinone and cytochrome C are mobile electron carrier. Means when ubiquinone is coming in complex 3 between cytochrome B and C1, it can act between the cytochrome A and A3, and when it is positioned accept an electron from cytochrome A during a transfer of electron from cytochrome A to A3, then it can push a H plus 2 from complex 4 as well. So such a way this particular whole process takes place. Now in this, the oxygen is terminal electron acceptor, and it is limited to the terminal stage of process, and it is known that the presence of oxygen is vital, and as we know the oxygen drives the whole process. It's remove hydrogen from the system from mitochondrial matrix to the perimitochondrial space, and it is also known that the oxygen is terminal electron acceptor as well as it is a final hydrogen acceptor from the mitochondrial matrix. Okay, so this way the process take place. Now, as we have discussed, there are 5 complexes. Complex 1, as we have discussed, where the electron passes and hydrogen passes from NADH2 to the FMN and through FMN to the ubiquinone. This is the complex 1, which is also known as NADH dehydrogenase complex or FMN NADH dehydrogenase complex, and it transfer electron from NADH to the ubiquinone. Ubiquinone located within the inner membrane, as we know, and ubiquinone is the mobile electron carrier between the complex 1 and complex 3, and also between the complex 2 and complex 3, as we have discussed. Now we remember this complex 2 can be inhibited. So the inhibitors or the inhibited by the complex 1 can be inhibited by the inhibitors like rotenone and amital. So these two are the inhibitor of the electron transport through the complex 1 or a inhibitor of a complex 1. Then complex 2 is there, which is known as a succinate dehydrogenase complex. It transfer electron from succinate to ubiquinone. It transfer electron from succinate to the ubiquinone or from FADH to the ubiquinone. It is also known as FADH2 dehydrogenase complex. FADH2 dehydrogenase complex. And there is no inhibitor of complex 2. Complex 3 is there, which is also known as cytochrome C reductase, or it is also known as cytochrome BC1 complex. This catalyzes the reduction of cytochrome C. Because of this, it is known as cytochrome C reductase by the transfer of electrons from ubiquinol to the cytochrome C1 and from cytochrome C1 ultimately to the cytochrome C. Because of this, it is known as cytochrome C reductase, and cytochrome C is the mobile electron carrier between complex 3 and complex 4. Cytochrome C is a small protein that is attached to the outer surface of the inner membrane that we have discussed. But we remember this complex 3 is inhibited by a inhibitor, that is antimycin. After this, there is a complex 4 which is known as cytochrome C oxidase. This cytochrome C oxidase is also known as cytochrome AA3 complex. And this cytochrome AA3 complex is there, which is also known as cytochrome C oxidase. This has a 2 copper center in transport electron from reduced cytochrome C, which is present on the outer face of inner mitochondrial membrane, to the molecular oxygen. It contains 4 units: cytochrome A, cytochrome A3, 2 copper containing proteins. Okay, so these 4 things ultimately forming a complex 4. Now it is known that this complex 4 is inhibited by cyanide and carbon monoxide. So if you want to check or inhibit the electron flow from complex 4, we can inhibit by using these inhibitors. When the electron transfer from one carrier to another via complex 1 to the complex 4 in the electron transport chain, they are coupled to the ATP synthase, that is complex 4, means all the complexes are coupled to the complex 4, 5, for the production of ATP from the ADP and organic phosphate. And how this is coupled, as we have discussed. So the number of ATP molecules synthesized depend on the nature of the electron donor. Oxidation of one molecule of NADH give rise to the 3 molecules of ATP, while FADH2 produces 2 molecules of ATP that we have discussed. Although the aerobic process of respiration takes place only in the presence of oxygen, the role of oxygen is limited to the terminal stage of the process. And because of this, we call it as a terminal electron acceptor, that is, particularly we can say the oxygen. Yet the presence of oxygen is vital. It drives the whole process by removing hydrogen from the system, that is from the mitochondrial matrix to the perimitochondrial space, and oxygen at the final hydrogen acceptor it also acts. Unlike photophosphorylation, where it is light energy that is utilized for the production of proton gradient required for the phosphorylation, in a respiration it is the energy of oxidation reduction utilized for the formation of proton gradient and breakdown of the proton gradient. So here oxidation reduction energy is utilized. Because of this, we call it the oxidative phosphorylation, oxidative photophosphorylation, phosphorylation. So it is this region that the process is called oxidative phosphorylation. So mechanism of ATP synthesis, if we explain, that is explained by the chemiosmotic hypothesis by the Peter Michel in the complex 5. And the energy released during the ATS is utilized in the synthesis of ATP with the help of ATP synthesis, that is complex 5. Complex 5, if we talk, as we have discussed in the previous diagrams, it contains, or has a 2 major component, that is F0 and F1. F0 is considered as a base piece and F1 is considered as a head piece. And F1, as we have seen, it is a peripheral membrane protein complex, and which is on the peripheral side of the inner mitochondrial membrane, on the inner mitochondrial side of the inner mitochondrial membrane, and it contains a site for the synthesis of ATP from the ADP and organic phosphate, whereas F0 is integral membrane protein. And F0 is located in the inner mitochondrial membrane, and that forms the channel through which the proton cross the inner membrane and moves from perimitochondrial space to the mitochondrial matrix. So the passage of proton through the channel is coupled to the catalytic site of the F1 component for the production of ATP. And for each ATP generated, 3 proton pass through the F0 from the inner membrane space to the matrix down the electrochemical gradient. It is sometimes written as 3, sometimes written as 2, but in NCERT it is written as 2. H plus when passes through F0, they carry out a ATP, 1 ATP synthesis. Okay. Now in this complex 5, which is also known as ATP synthase or ATPase, that carry out, we can say, the oxidative phosphorylation, and it can be inhibited by inhibitor, that is oligomycin. It can be inhibited by the oligomycin. Okay, so this is what about the particularly we can say the electron transport chain. Let's discuss some of the questions. The number of complexes involved in ETS are. So as we had discussed, the number of complexes in ETS are the total five complexes. Actually, in electron transport system, only four complexes are involved, but one complex, that is complex 5, is involved in what oxidative phosphorylation. The number of mobile carriers involved in ETS are, obviously, as we have discussed, ubiquinone and cytochrome C are the mobile electron carrier. So answer is going to be 2. The mobile carrier between the complex 3 and complex 4. So the between complex 3 and complex 4, cytochrome C is located, and which is present on the outer surface of inner mitochondrial membrane. The mobile carrier between complex 1 and complex 3, obviously this is going to be ubiquinone, because ubiquinone carry out a transport of electron between these complexes, which is also known as coenzyme Q. The terminal electron acceptor is respiration is, in respiration terminal electron acceptor is oxygen. Okay. So this is what about the electron transport system and oxidative phosphorylation, and how NADH2 and FADH2 get oxidized, and NADH2 gives 3 ATP and FADH2 gives 2 ATP that we have discussed. I hope you understood well. Thank you. Thank you everyone.